Namaste. I'm Jivin Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I'm going to discuss about the topic culture media and what are the different culture media. This topic belongs to the microbial nutrition and it is related to the fundamentals of microbiology subject. So how we can cultivate the microorganism. For the support of microbial growth, we prepare some specific formulations. Okay, and these supporting, which will enhance, which will support the growth of the microorganism, is known as the culture media. And the culture media can be solid, it can be in liquid, it can be semi-solid. So the media can be solid, liquid, and semi-solid. And the solid media are usually solidified using the agar, and the liquid media components are dissolved in the water, and they are uh, dissolved in water, and agar is not used, and the media is sterilized. The semi-solid media is a medium which has been added a gelling agent. Means agar like gelling agent is used, but the concentration of the agar is lower. Another is gelatin. Gelatin can also be used, and silica gel can also be used as a uh, solidifying agent in the semi-solid media. There are different types of media are there, in which some of them are defined and some complex. Okay, and in what are the chemical defined and complex media? Chemical defined media are the exact chemical composition are the media whose chemical composition is known. Okay, so if we know any media whose chemical composition is known to us, then it will be known as the chemically defined media. Example is the minimal media used in bacterial genetic experiment is known as the chemical defined media. Now, if we talk about the complex media, the exact chemical composition is not known. The media whose exact chemical composition is not known is known as the complex media, and they often consist of plant or animal extract such as soybean meal, milk, protein, etc. So they are not known. We do not know how much composition can be there, and they include most routine laboratory media like tryptic soya growth. Next type is the synthetic or defined media. Next, this is a new, also a variant of the culture media, and all in the synthetic or defined media, all the components of their concentration are known. So, if we know the chemical composition of any media, they are known as the synthetic media. For example, if we want to grow cyanobacteria, if we want to grow especially E. coli, then the caption, the defined media composition is known to us. Next is the complex media. The complex media which contain some ingredients of known composition or concentration are known as the complex media. Means some of the components ingredients are known, they are known as the complex media. Okay. For example, nutrient growth and uh, triple soya growth and conic agar. So these are the example of the complex media. Some of the media components which we can say here about that peptone is also a mm, media component, and protein hydrolysates prepared by partial digestion of various protein sources. It is in the extracts, aqueous extracts, usually beef. And similarly, agar is also a component. Peptone is a component. Some extract like beef and yeast are also component, and agar is also a component. Sulfated polysaccharide, which is used in solidification of the solid media. This is the example of the uh, solid media, and they are used in different proportion in different experiments. Some we can also uh, expand our knowledge by knowing some more types of media. The general purpose of media uh, is to support the growth of many microorganisms like tryptic soy agar, and in this media is a media which have the general purpose media, and they are supplemented by blood or other special nutrients. The example include the blood agar. Selective media is the next type. It favor the growth of the some microorganisms and inhibit growth of others. It favor the growth of some microorganisms and it inhibit growth of others. And McConaughey agar is also a is a bacterial media, and it is usually used for the select the gram-negative bacteria, and it inhibit the growth of gram-positive bacteria. Differential media is also a type of the media. Differential media distinguishes between different group of microorganisms based on their biological characteristics. Like blood agar is a differential media, and hemolytic versus hemolytic, non-hemolytic bacteria. And the Mekong agar is also an example of the differential media, lactose fermenter versus non-fermenter. So these are the some examples of some bacteria, media, some microorganism which we can use are of different types. For example, some are mm, bacterial media and some are for the fungal media. If we talk about the bacteria, so there are different types of culture media which we have discussed here. Now we can also discuss about the fungal media. What are the fungal media? Fungi also need some specific culture media. For example, we can use some energy source, some sucrose source, and some gelling agent. They are also divided into solid, semi-solid, and liquid media. Solid media is known as the solid media, and the liquid media is known as the broth, and semi-solid is known as the semi-solid, in which the concentration of agar is lower. And it is gel-like medial composition. So, if we talk about the different types of fungal media, then fungal need some energy source, some sugar source. Okay. So, on the simplest media is the potato dextrose agar for the fungus, in which we use potato infusions, and from for these potato infusions we can use the potato and along with some amount of the dextrose and also agar. then this will uh, after autoclaving, the media will be prepared with the dextrose agar. Similarly, we can also use the dextrose agar in which peptone uh, and dextrose and agar are mixed in different proportion for the preparation of STA. So this is the media which is used for the fungal. So in this video, I discussed about some different type of media for bacteria and fungi. So what kind of question which can we ask? According to this lecture, the question which can arise: explain microbial growth media and their types. So this question can be asked. Similarly, examiner can ask you some questions uh, about some examples of different type of media. For example, they can ask you what is the example of the selective media, what is the example of the differential media. So these questions can be asked. So my lecture is over here. If you have any query, you can ask me. So thank you very much.